This is a production of Cornell University.
Okay.
Consumption on salads to making jams and baking. They're really interesting for their nutritional qualities and their unique specialized metabolites. Breeding programs haven't been developed for these. There aren't really a lot of cultivars. They're mostly still land races or just farmer preferred accessions. So they have not been improved for large scale agricultural production or growth. So in the Van Eyck lab, we are particularly interested in ground cherries and golden berries, which are among the most agronomically important Physalis crops, making up the second largest fruit export of Colombia. On the left in this photo, we have the ground cherry, which is Physalis griseia. And then on the right, we have the golden berry, which is Physalis peruviana. So a major challenge in adopting these species as agronomically important crops in North America and also globally is pest pressure. In a citizen science project performed by the Van Eyck lab, which involved people from across all of the US, more than 43 states participated. We saw a lot of insect pressure from this Lima datura, which is the three-line potato beetle. The adults cause damage on the leaves of the plants, and the larvae also would hatch and cause damage on the leaves as well. But we also noticed through the citizen science program that there seemed to be difference in presence on different accessions and in different species of Physalis. And we really want to take advantage of that natural variation if we can in pre-breeding in order to allow these crops to be adopted more widespread. So in preliminary insect bioassays, we saw differences in insect interactions on different accessions or genotypes of Physalis griseia, which is the species that I will mostly be talking to you about for the rest of this presentation. There was a difference in the number of eggs laid on accessions, as well as a difference in the weight gain of larvae after hatching. And this work was conducted by Julie Davis in collaboration with the Van Eyck lab. A untargeted metabolic analysis of different accessions of both ground cherry and golden berry revealed a lot of variation in metabolite profiles. So this heat map shows different expressions or different presence levels of metabolites. And you can see there's a lot of variation both between accessions of the same species and between species. So that's really interesting and leads us to believe that this difference in metabolite presence might be contributing to the variation we see in insect behavior with these plants. So in particularly, we're interested in a metabolite known as withanolides. The reason we're interested in these is because they have been reported to be insect antipedants in other species, but there are more than 100 withanolides that have been identified in Physalis, and very few of them have been characterized. These compounds are also unique because they also confer human health benefits, unlike other insect antipedant compounds such as cyanic glycosides and cassava that can cause issues for human health. So that makes them really interesting to look at as a target for pre-breeding and crop improvement. So what we want to do is use a push-pull-protect method to manipulate flux in the withanolide biosynthetic pathway. And the way we will do this is by increasing precursors in the pathway, by increasing the pull of flux through the pathway by increasing rate limiting enzymes, and by protecting the pathway from competing pathways. So we want to knock out enzymes that lead to competing pathways in order to get to increase the expression of withanolides as much as possible. And we can use CRISPR-Cas9 methods to generate mutants that have these particular traits that we're looking for. So knockdown mutants have been generated in the Van Eck lab, targeting these two genes that I have highlighted here, Cas and 24-iso, which are key genes in the biosynthetic pathway. Unfortunately, the Cas knockdown mutants were not successful because we actually saw lethality in these knockdowns. However, the 24-iso knockdowns were successful, and we have been able to recover a few mutant lines using this method. In an LCMS analysis of leaf extracts from both the knockout of the 24-iso mutants and our wild type plants, we do see a lot of metabolic variation, metabolite profile variation. And what's really interesting is we looked at withaferin A, which is just one withanolide compound found in Physalis, which we have a standard for, unlike the remainder of the 100 plus withanolide compounds. And what we found was that in the wild type, there are two peaks for withaferin A isomers, one that is quite large and a smaller one. But in the knockout mutant, we actually saw both peaks were very greatly reduced, and that's the red here. So we took our knockout lines and conducted insect bioassay studies using Lema desterophila, which is the beetle I mentioned earlier. We didn't see a lot of differences in larval leaf area consumed, but we did see a statistically different amount of adult female leaf area consumption using an ANOVA 2-factor test. So in the future, we want to, since we had issues with the knockout for the Cas gene, we actually want to generate mutants that are knocked down. So instead of targeting the coding sequence with Cas9, we will target the promoter sequence. And this will allow us to generate mutants that have differential expressions of withanolides, hopefully. And I am currently working on generating these knockdown mutants. And I am also starting to create more knockout mutants targeting different genes in the pathway. And the overall goal for this is to create a library of functional mutants that we can use to identify the particular metabolites that affect insect resistance. And we will conduct further insect bioassays to test the effects of these different things and hopefully identify a particular withanolide. And if we can't, then we are also interested in looking at further metabolites that might be contributing to this variance in insect resistance. And with that, I would like to thank my special committee, the entire Van Eck lab, and all of our collaborators who are wonderful. And thank you all very much, and I will take any questions.
Thank you for the wonderful talk, Savanah. That was very interesting. We already have one question in the chat, but also feel free to raise your hand or unmute yourself. I'm going to read the question from the chat, which is from Amy. She asks, what is the benefit of targeting multiple upstream genes instead of just the first committed step?
Okay. So instead of just targeting 24-iso, which we think is probably the first committed step, we are targeting upstream genes because we don't know if the products of the upstream steps are then also utilized in downstream steps because this pathway isn't very well characterized. And because we need to think of metabolic pathways as networks instead of just single flows. If that makes sense. Okay.
I had a
Yeah.
I had a question about targeting the upstream steps and whether your problems are going to be with the brassinosteroid biosynthesis, that if you knock that out or overexpress brassinosteroids, you're likely to have either dead or messed up plants, and how are you going to deal with that?
That is something that we do think is the issue, and that's kind of what we think happened with creating a knockout version of the targeting Cas. And that's why we also want to generate these knockdown mutants, because hopefully by targeting promoters and lowering expression, we can still recover mutant lines without experiencing that lethality. Because that is something that we've considered, because the brassinosteroid pathway is so intricately linked to this one. So that is definitely something we've considered.
Okay. Jean-Luc Yannick asked, can you say more about the possible human health benefits of withanolides? Any danger of there being too much?
So in a lot of studies, they've been looked at as anti-tumor treatments, and also as anti-inflammatories, and they have a lot of antimicrobial properties. However, I don't know if they found an upper limit where there might be too much, and that's definitely a good question, especially if we do find that these are useful in reducing or increasing insect resistance. We would want to find those upper bounds, but I don't, as of now, know if there are upper bounds.
Okay. We have another question from Lauren. She's asking, is there a relationship between adult insect oviposition preference and leaf damage? If I recall correctly, you showed that those were two metrics of resistance. How do withanolides affect both phenotypes?
So we are actually investigating oviposition, and I'm not able to share that data at the moment. But we do expect there to be a correlation between oviposition choice and larval damage on the leaves. It just makes sense, right? There have to be eggs to hatch out for there to be larvae on the leaf to damage the leaf. So that is something we will be investigating further.
This has been a production of Cornell University, on the web at cornell.edu.